Hello everyone, welcome back to my channel Xgene and proteinX. In this tutorial video, I'm going to talk about overview of the mycoplasma and detection of the mycoplasma in cell culture. So as we know that most of the cells are contaminated by the mycoplasma. So we need to know about the mycoplasma in details. So here I will discuss about the mycoplasma in details. So watch this video till end. Let's begin. So first, the mycoplasma is the genus of the bacteria, and is a class is the molecules, and cell wall is absent. So this is the main difference in between the mycoplasma and the bacteria. The bacteria has the cell wall, but mycoplasma has no cell wall. And this is the structure of the mycoplasma. So it contains lipoprotein membrane, soluble protein, ribosome, DNA, and the soluble RNA. So here in this figure, as you see that in the bacteria it has the cell wall, but in the mycoplasma there is no cell wall. So this is why the antibiotics normally we use for the penicillin and the streptomycin for the as antibacterial in the cell culture, they're gonna work for the bacteria, they're gonna impair the synthesis of the cell wall. But this antibiotic, it does not work in the mycoplasma because it does not have the cell wall. So next is the different species of mycoplasma found in cell culture. So here I'll talk about different 5 species are isolated in 90 to 95% of contaminated cell cultures, and those are Mycoplasma aeruginosa, M. fermentans, M. oral, and M. hyaluronis, and also Calorimetric light Y. So these 5 different species of the mycoplasma are mostly found in the 90 to 95% of the contaminated cell culture. So next is the mycoplasma contamination. So the mycoplasma that contaminate cell cultures come from 3 principal sources, and those are cells donated by the other labs. So when you start culturing the cells, and when we first begin the cells culture, so that cells might be the already contaminated by the mycoplasma. Number 2 is the porcine trypsin, and this is actually the extracted from the pancreas of the pigs. So there is a tendency of this kind of trypsin contaminated by the M. hyaluronis, this kind of species of the mycoplasma. Number 3 is the bovine sera, and also bovine sera it might be contaminated by the M. arginine and L and A light Y. So these 2 species can be contaminate this bovine sera. So once an area has become contaminated, the mycoplasma can spread by aerosol droplet dispersion, and then it can contaminate the incubator, the pipettes, and even the cell culture hood. So it's gonna influence your cell culture completely. And next is unlike bacteria or fungi, mycoplasma do not cause consistently observable alteration in cell culture such as rapid pH change or culture turbidity, because we know that in the bacteria culture, in the bacteria contamination, the cell culture supernatant, it can turbid very easily, so we can identify the bacteria. But in case of the mycoplasma, we cannot identify because it is very much tiny, and there is no pH change or no supernatant change, so it is very hard to detect. And also the mycoplasma can have disastrous effect on eukaryotic cells. At the beginning, it has very less effect on the cells. Cells can grow easily, no problem, but later on over time mycoplasma it influence cellular growth, proliferation, protein synthesis, and even it has the tendency to the viral infection. So these are the outcome of the mycoplasma contamination. So here the visual observation under microscope. So although it is very hard to identify in the cell culture, but you can also identify if you can observe very well. So here is a cell, normal mammalian cell, and this is the 10x picture. So if you magnify this picture, for example, this area I can magnify to 40x, alright, and then you can see here very clearly the background is very clear, so cells are not contaminated by any other kind of contaminant. So here as you see this kind of round shape, and this round shape you don't misjudge, because these round shapes are the multiplying cells. When the cells start divide, these cells looks like the round shape. So this is normally the round shape cells. These are the cells, right? No problem. But here as you see, if you see very clearly here, so this red arrow, this showing this is like triangular shape, and this is definitely not the cells. So this is the contaminant. So this is the actually this is the mycoplasma, because in the beginning this number of the mycoplasma you can see very less, but over time you can see it can grow, it can increase in numbers. So you can observe under microscope by this way. So next is the detection of the mycoplasma. So here there are different methods that you can detect the mycoplasma. There are many kinds of kits available in the market. So first is the direct growth on growth agar, specific DNA staining, RNA labeling and enzymatic procedures, PCR, ELISA, and calorimetric mycoplasma detection assay. But I will discuss just something little about three or four process, not that much, but just I want to give you some basic idea that how this kind of kits works actually. And also the protocol also available in the manufacturer's manual, so if you use it, so you can follow that protocol. So first I want to talk about the specific DNA staining. So if you stain your cell by DAPI, we know that the DAPI it can stain the nucleus, right? So here as you see the DAPI staining. So here as you see here, these cells are very much clear. There is no background noise. So this is the cell's nucleus. But here as you see in the background there are a lot of noise. So it means that DAPI it stained the other contaminant nucleus. That might be the it's obviously the mycoplasma, because mycoplasma grow slowly. So these are the mycoplasma contamination. Alright. So next, and also the specific DNA staining procedure are relatively quick and are easy to perform. So next is the PCR. The PCR method also fast and inexpensive. So for that also you need to use the kit. So everything available in the kit. So first you need to prepare the sample, means that your supernatant, and in that sample preparation you need to also use your primer and everything else according to the manual, and also it amplify 16S rRNA gene, and this 16S rRNA gene it most commonly found in the Mycoplasma species in the cell culture. Alright, and then you will get the result, and that result can be visualized in the immunochromatographic strip. So this is a different kind of process, so I'm not gonna discuss here in details about it. So this is just the basic process about how it works. And next is the enzymatic procedures. So here also you need to prepare the sample like supernatant, and then you need to lyse this sample, and after lysis the mycoplasmal enzymes it release, and that mycoplasmal enzymes it gonna react with the substrate. That substrate is provided by the kit, and when this substrate it will bind with the mycoplasmal enzymes, then it catalyze the reaction from the ADP to ATP, and then this ATP it transferred through the luciferase enzyme, and it gonna gives the light signal, and through this light signal the ATP can be measured, and this ATP also this ATP need to be measured before. So this ATP can be measured before addition of the substrate and after addition of the substrate. Alright, and then this ATP measurement, it gonna shows the presence or absence of the mycoplasmal enzymes. So it means that mycoplasma present or not. Next is a calorimetric mycoplasma detection assay. So this is the most convenient process. People normally follow this kind of process. So here also need kit. This kind of kits are also available in the market. So first, the kit contain the cells, and that cells contain toll-like receptor 2 or TLR2, and this receptor it gonna recognize the pathogen. So this is normally is known as the pathogen recognition receptor. Alright, and presence of the mycoplasma, this toll-like receptor, it activate NF kappa beta pathway and other transcription factors, and later on also it secrete ACAP or secreted embryonic alkaline phosphatase. Mainly this is by the mycoplasma, and it gonna secrete in the supernatant. So this ACAP activity is readily detected by the color, observation of the color. This color is a purple or blue. So if you get purple or blue color, so that means there is a presence of the mycoplasma. So this is very much easy process, and easily you can detect the mycoplasma. This is very convenient. So this I discuss here different process how to detect the mycoplasma. So I think it will be very much easier process to detect. In my next video, I'll talk about the how to get rid of the mycoplasma from the cell culture. So this is all about the overview of the mycoplasma and the detection. So I hope this video will be helpful. If you like this video, kindly hit the like button, share it, and please subscribe my channel. And if you have any queries, kindly write in the comment section. Thanks for watching.